Hello, I'm Dr. Stephen Pike from the University of Michigan. I'm an investigator on the HOPE-B clinical trial program, and I'm going to give you a summation of the presentation I gave at this year's ISTH. Following several decades of preclinical and early phase clinical studies, the platform of gene therapy that demonstrated the most favorable safety and efficacy profile was a liver-directed approach by which a functional copy of the gene for either factor VIII or factor IX, what we call the transgene, is delivered using adeno-associated virus vectors. And the advantages of this approach is that they can efficiently target the nuclei of liver cells, yet the delivered DNA primarily remains outside of the chromosomes as a circular DNA. So this limits the risk of adverse consequences of transgene elements integrating into the patient's own DNA. However, we know that rare integrating events can still happen, and considering the vast number of vector particles that are delivered and the large number of cells that are transduced, integrating events are likely to occur in many cells. So the question then is, do these integrating events pose any risk to the health of the recipient, and how would this be manifest? One concern is whether rare integration events could trigger cancerous transformation of cells. Now, when targeting the liver, the main concern would then be hepatocellular carcinoma. However, the hemophilia population carries unique risks for this cancer already. Hepatocellular carcinoma is strongly linked with prior infection with hepatitis B and C viruses, where these are associated with 80% of all cases of hepatocellular carcinoma. Gene therapy trials to date have not excluded patients with a history of hepatitis B and C, provided they have no active disease and have gone through some treatment to eradicate the hepatitis C virus. Most cases of hepatocellular carcinoma occur among those patients with a history of Hep B and or Hep C with advanced fibrosis. So to this end, patients are screened for their liver health before being deemed eligible for these gene therapy trials. However, there's a critical issue in that hepatocellular carcinoma risk is reduced, but not completely eliminated with hepatitis C eradication. Furthermore, additional risk factors such as non-alcoholic fatty liver disease, non-alcoholic steatohepatitis can also influence that risk. So I presented a case of hepatocellular carcinoma in a participant in the HOPE-B gene therapy trial for Hem B. This uses AAV5 vector, and it contains the hyperactive PADOA factor IX transgene. This was a 69-year-old gentleman who had multiple risk factors for hepatocellular carcinoma, given that he had a history of both hepatitis B and C, contracted more than 30 years ago, but had undergone hepatitis C eradication treatment. He was a prior smoker and had some alcohol consumption history, as well as a family history of cancer. Yet, he still met eligibility for participation in the trial based on his liver health evaluation. All participants in the trial had baseline liver ultrasounds before starting treatment. 1 year post-gene therapy, a per-protocol follow-up liver ultrasound detected a liver mass, and a biopsy confirmed hepatocellular carcinoma. At the time of a surgical resection, a second lesion was identified, and he was treated with surgery to downstage his presentation. And then the second lesion was treated with chemoembolization. The participant is in ongoing discussions regarding suitability for liver transplant. The participant had sampling of both the tumor specimen and adjacent normal liver tissue that was then subjected to extensive genomic analysis. So this allowed investigations to determine the percentage of cells that had integration events and to evaluate for the proximity of these integrations in or near known oncogenes. Well, what was observed was that, as expected, integration events were very low, as reported previously with AAV gene therapy. About 0.027% of liver cells. And notably, there was no dominant integration site within the hepatocellular carcinoma sample. Rather, multiple structural chromosomal variants were observed in the hepatocellular carcinoma sample, large chromosomal rearrangements, as well as mutations in oncogenes, all that were well described to occur in hepatocellular carcinoma independent of gene therapy. So this provided solid evidence that the hepatocellular carcinoma that developed in this patient was unlikely related to the gene therapy treatment and rather attributable to the patient's underlying risk factors. Short-term and long-term follow-up in gene therapy is critical to reporting the safety of this platform of therapy to the hemophilia community. Short-term modifications to this protocol is that ultrasound monitoring will be increased to twice yearly, but not because any link to the gene therapy was identified, but more so because we recognize the pre-existing risk factors of the participants in these trials remain significant. Longer term, it'll be important for all participants in gene therapy to be followed over the course of a lifetime to fully inform on both the safety and efficacy of this new platform of therapy for hemophilia.